Hello guys, today we'll discuss about transport mechanisms across the cell membrane. Question may be asked as enumerate different transport mechanisms across the cell membrane or transport through the cell membrane. Let us start with the answer. First of all, what is the importance of transport of substances? All the physiological activities of any cell, it depends on transport of various substances. Like suppose there is a cell. So substances like nutrient and oxygen, they enter inside the cell, and waste products and carbon dioxide, they exit outside the cell, and this depends on various transport mechanisms. Now, transport mechanisms are divided in mainly three types. Number one, that is passive transport, which is also known as downhill movement. Means here transport of substances along the gradient. Gradient means from higher to lower. So along the gradient of concentration, along the gradient of pressure or electrical. Okay. So substances are transported along the concentration or pressure or electrical gradient, whereas in active transport, this is the transport in which substances are transported against the gradient. This is also known as uphill movement. Means here substances are transported from low concentration to high concentration. In passive, that is from high to low. Okay. Here low to high, or pressure gradient or electrical gradient. Okay. Third variety, that is special category of transport, one of them that is vesicular transport. Now, passive transport, that is also known as diffusion. Other very important name for the passive transport is diffusion or downhill movement. It is again divided in two types. Here diffusion, we have discussed transport of substances along the gradient, and it is further divided in two types. Number one, that is simple diffusion, and number two, that is facilitated diffusion. Now, starting with simple diffusion. What is simple diffusion? If we are defining simple diffusion, that is random thermal motion of molecule, random thermal motion of molecules in a liquid or gas, in a liquid or gas. What happens because of this movement? There is distribution, redistribution of the molecule. Redistribution of the molecule, and this takes place till the state of equilibrium is achieved. It is like, if I am giving you example, like suppose you have a glass of water, you are adding either salt or sugar in it. So what happens? There is random thermal motion of the molecules in a liquid or gas, and that is for the redistribution till the state of equilibrium is either sugar or the salt that is distributed equally. Okay. So the movement stops when the state of equilibrium is achieved. That is known as diffusion equilibrium. Diffusion equilibrium. There is one law, fixed law of diffusion, that denotes, here this is J is equal to dA into C1 minus C2 divided by T. So here you can see J, that is rate of diffusion. So rate of diffusion depends on, rate of diffusion is equal to, this is constant, diffusion coefficient. A is the surface area or cross sectional area. C1 minus C2, that is concentration gradient. Concentration gradient. And T, that is the thickness of membrane. So these are the factors, they affect diffusion. So diffusion of the molecules across the membrane, it depends on various factors. Number one, that is lipid solubility. If the substance is soluble in lipid, there is no membrane, so they can easily pass. And which are the substances? Here examples are oxygen, carbon dioxide, alcohol, steroid hormones. Okay. Weak acids, bases, they all are lipid soluble. Second is water soluble. If substance is water soluble, then they cannot pass through lipid bilayer. They will pass through protein layer. They have protein channels for the transport of the substances. These protein channels, again, they have two characteristics. Number one is selective permeability and gating mechanism. Selective permeability, one characteristic, and gating mechanism. Other factor is electrical charge. If the charge of the substance is negative, it will easily pass from negative to positive side. Size of the molecule, size is small, can easily pass, and selective permeability of the membrane. Okay, so these are the factors, they affect simple diffusion rate. Now, as we have discussed, protein channels, they have two characteristics. Number one is selective permeability, second is gating mechanism. Selectively permeability means the channel is permeable for particular substance only. Like for example, if the channel is sodium, it will allow sodium ions to pass only. So selective for sodium ions. As like sodium channel size is 0.3 to 0.5 nanometer. Okay. Then and these channels are strongly negatively charged. Okay, potassium ions channel, that is again of size different, 0.3 to 0.3 nanometer, 0.3 by 0.3, and it is not negatively charged. Second is gating mechanism. So this gating mechanism, that is important for maintaining or regulating permeability of the channel. Means when the gates they are closed, substance will not pass. So here gating mechanism, according to gating mechanism, channels are of two types. Number one is ungated channels. There are no gates. They are continuously open or continuously closed. And second are gated channels. And these gated channels again are of three types: voltage gated channels, ligand gated channels, and mechanically gated channels. Voltage gated channels, as the name suggests, these channels they are opened or closed by change in the voltage. Change in voltage. Okay, change in voltage. Like for example, if it is a sodium channel. So if we are talking about sodium channels, gates for the sodium channels are located outside, and these gates are closed till there is strong negative charges inside the membrane. Means when the inside of the membrane loses its negative charge, then the gate will open. Second is potassium channel. So potassium channels, their gates are present inside the membrane, like this here. This is the gate. And this gate is also open when cell membrane becomes positive charged inside. So another important characteristic of potassium channel is this: potassium channels are slower to open as compared to sodium channel. Yes. Second variety of channels are ligand gated channel. What do you mean by ligand? Ligand is any substance like hormone or any chemical. So when this substance, suppose this is a channel, when here is a place for binding of the ligand. So when the ligand binds with this channel, there is opening of the channel. Channel is not for the ligand. For example, we all know acetylcholine gated channel. So when acetylcholine binds with this site, there is opening of the channel, and the channel is either for sodium or any other substance. Okay, here it is for sodium when we are talking about acetylcholine. So when ligand binds, channel opens and it will allow the transport of the substances. Yes. Third variety of channels are mechanically gated channel. Means these channels, they are opened or closed by mechanical change, stretching or any sound wave. For example, Pacinian corpuscle. By pressure, this channel they get opened up. Hair cells present in the ear, in the inner ear organ of Corti, they are also mechanically gated channels. Next type of diffusion, that is facilitated diffusion, which is also known as carrier mediated diffusion. Okay. Now here the substance is large enough. The substance is water soluble and large. Two things are there: water soluble, so it will not pass through lipid bilayer, and the substance is large. For example, glucose, so it will not pass through the lipid bilayer. And here this substance, because it is large, it requires one protein, carrier protein. Okay, so this transport is known as carrier mediated diffusion. I'll show you the example. You can see here, this is a carrier protein. When the substance binds with this carrier protein, then and then only it can enter inside the cell. Important here is, because it is diffusion, so facilitated diffusion means from high to low. So two things are required. Number one, gradient, high to low, and number two is carrier. In simple diffusion, only gradient is required. So this is the difference between simple and facilitated diffusion. In facilitated diffusion, not only gradient is required, but the carrier molecules are also required. Okay. So this is example. One of the example is transport of glucose. For example, suppose if we have taken large amount of glucose, that will not enter inside the cell, not all the glucose. So glucose transporters, they are mainly present in the intestinal epithelium as well as in the RBCs, muscles and adipose tissue. Now, here one clinical application is there. Suppose you can see here, this is your carrier protein for glucose, okay, and that is required for the transport of glucose. So what happens in diabetes mellitus, this carrier protein requires insulin also. So if insulin is not there, then this carrier proteins, they will not allow transport of the glucose inside the cell, and this is the reason why glucose is more outside the cell, and outside means increased sugar or glucose in blood. Okay, because it will not enter mainly inside our skeletal muscle cell, because this glucose transporter that requires insulin. Okay, different types of carrier proteins are there. Basically three types of carrier proteins. One, that is uniport. Uniport means this carrier proteins, they cause transport of one substance, only single type of molecule. Single type of molecule transported. Symport means two molecules are transported but in the same direction. Symport means in same direction. And antiport means in opposite direction, like one enters, one exits. So three types of carrier proteins are present. Next is channel mediated diffusion. There are ionic channels which are located in the cell membranes, and they use ions, and they are specific for that particular ion. For example, if we are talking about potassium, so potassium channel, that is having 4 similar protein subunit, and they are required for the transport of the potassium. Ligand gated cation and anion channels also, they have different proteins. Now there is one clinical significance. Chloride channels are also there, that is channelopathy. What is this channelopathy? Channelopathy. When there is mutation of gene for the channel. For this channel we have a gene for the formation of the channel. So if there is mutation for the ion channel, gene is mutated, then that results in the channelopathy. Okay? And membrane permeability depends on the channel, so the membrane permeability is affected. Now various factors, they affecting rate of diffusion. Number one is permeability of the cell membrane, which includes thickness of the membrane. If the membrane is thick, rate of diffusion decreases. So this you can see inversely proportionate. Okay, inversely proportionate. Then temperature, increased temperature, increased rate of diffusion. So directly proportionate. Lipid solubility, increased lipid solubility, increased rate of diffusion, directly proportionate. Protein channels, more the protein channels, more will be the diffusion rate. Okay, so that is directly proportionate. Then size of the molecule, if the size is small molecules, then rate of diffusion is fast. Size of ion is, so this is, you can see, inversely proportionate. Then size of ion, again inversely proportionate. And charge of ion means if the charge is greater, then transmission is less. Ca plus 2, the transmission rate is less than plus 1. Okay. First factor we discussed, that is permeability of the membrane. Second factor required for the diffusion, that is concentration gradient. Rate of diffusion is directly proportionate to the concentration gradient. If more concentration this side, less this side, then diffusion is fast. Electrical gradient are again directly proportionate from negative to positive side. Pressure gradient, more pressure to less pressure. So 4 factors: permeability of the membrane, concentration, electrical and pressure gradient. Now, what is the difference between simple and facilitated diffusion? As we all know, in simple diffusion we don't require carrier protein. So that is one of the important difference, carrier proteins. Here, because of carrier proteins, this facilitated diffusion is specificity. Carrier molecules are specific for the substance. Here. Then second characteristic is saturation. Now what is this characteristic saturation? So if we are talking about here, you can see saturation. What is the meaning of saturation? Here suppose this is the concentration of substance and here it is rate of diffusion. So what happens in simple diffusion? In simple diffusion, as the concentration of substance increases, you can see this is the line for simple diffusion. As the concentration increases, rate of diffusion increases. And second one, this is for facilitated diffusion. Initially you can see as the concentration of substance increases, rate of diffusion increases, but when carrier proteins all are completed or all are saturated, now there is no more increase in the rate of diffusion in spite of increase in the concentration of substance. The example is suppose you have taken a large amount of glucose, but if all the carriers are saturated, then now this glucose cannot enter inside the cell. So this is second important characteristic is saturation. Third is competition. Here there is a carrier protein in facilitated diffusion. So there is a competition between substrate and any other substance which can competitive substance which is having competition to bind with the carrier molecule. So this is all about our diffusion, that is passive transport. Next is active transport. As we all know that in active transport, substances are transported against the gradient. Against the gradient means from lower to higher concentration. So it is known as uphill movement. Suppose this is the cell. Inside the concentration is less, outside is more. Still the substance, they will be transported. And when we are going against the gradient, we require energy. So active transport requires energy. Okay, and this energy comes from either directly from the ATP or from other source. If it comes directly from the ATP, this transport is known as primary active transport, and if it is from other source, that is secondary active transport. Various substances, they can be transported from active transport. They are your sodium, potassium, calcium, H+, chloride, iodide, okay, glucose, amino acid, urea. You might have questioned that, madam, the sodium, potassium were transported through passive transport also. So both the types of transport are possible for the substances, active as well as passive. If there is a gradient, then there is active. If there is a gradient, then there is passive transport. If it is against the gradient, then there is active transport. So both the varieties of transport are present for different substances. For glucose also, active as well as facilitated diffusion, both are present. Now, primary active transport. Here, primary active transport means energy is required because it is active transport. But here this energy comes directly from the ATP. So it is known as primary active transport. One of the important example for primary active transport is sodium potassium pump, which is also known as sodium potassium ATPase. What is the structure? Let us first discuss about the structure. This is the sodium potassium ATPase. This you can see here, this has two subunit. It is like this. One is beta subunit and other is alpha subunit, like this. Okay, so this is alpha, this is beta. Here, alpha subunit, the molecular weight is 1 lakh. Beta subunit, the molecular weight is 55,000. Functions of beta subunits are not very much known. Alpha subunit has 6 binding sites. 6 from which 3 for sodium inside the cell membrane and 2 for potassium outside the cell membrane and 1 for ATPase. So this pump gets active when 3 sodium ions they bind inside, 2 potassium binds outside the membrane. Now, what is the mechanism of action of this pump? When 3 sodium ions they bind inside, ATP binds with this ATPase side and 2 potassium binds outside. What happens? This pump gets active and it causes movement of 3 sodium ions out and 2 potassium ions in. Okay? So what happens? As we all know, sodium is more outside the cell, potassium is more inside. So against gradient, sodium moves out and potassium moves in. And it requires energy which comes from ATP. What are the functions of this pump? One, that is electrogenic activity of the pump. Why electrogenic? Because this pump causes movement of 3 positive outside and 2 positive inside. So 1 positive charge moves outside. Okay? So cell membrane remains negative inside because of this activity of the pump. Second is it maintains cell volume. Why? Because inside our cell we have proteins. These proteins are negatively charged and they have tendency to attract positively charged substances inside. So what happens along with sodium ions because they are more outside, so along the gradient through diffusion this sodium have tendency to enter inside, and when sodium enters water also enters inside. So the cell gets swollen because of entry of the positive ion, because through diffusion the sodium, because they are more outside, they will enter inside and the cell gets swollen. But when the pump is active, what does it do? It causes movement of sodium outside. It is like we have a heavy rain outside and the water trying to move inside our house, and we are actively moving the water outside. So 3 sodium ions are moved out, 2 potassium ions they enter. So cell volume gets maintained. Okay. Now applied aspect here is the pump inhibition. When the pump gets inhibited, because this pump requires 3 sodium, 2 potassium and ATP. When all these 6 they bind, then only this pump gets activated. If any of the substance is not present, then the pump cannot be activated. That is one inhibitory factor. Second, it requires temperature. Very low temperature also causes inhibition of the pump. Third, because ATP comes from aerobic glycolysis, so oxygen is required. So first is substances. Second requirement is temperature. Third requirement, that is oxygen. Fourth requirement, sometimes some poisons, metabolic poisons like 2,4-nitrophenol, it also inhibits the ATP. So the pump activity is inhibited. Fourth factor is excitation. Some hormones like thyroid hormone increases activity of the pump. Insulin, aldosterone, G-actin, they all stimulate the pump activity, and dopamine inhibits. So inhibition is by dopamine, activation is by your thyroid hormone, insulin, aldosterone and G-actin. Clinical significance. One of the clinical significance is in case of heart failure, a drug used, that is digitalis. How it works? It increases contractility of our heart muscle. How? Because you can see this is our heart muscle cell. So digitalis, it binds with the extracellular part of the alpha subunit. We all know this is alpha. So it binds with the alpha subunit. Okay, and it affects dephosphorylation step. So it inhibits the pump. In short, you can remember it inhibits the pump. So when this pump gets inhibited, it inhibits sodium potassium pump to increase intracellular sodium ion, because what is the activity of the pump? The pump causes 3 sodium moves out and 2 potassium in. So when the sodium ion concentration is more inside, as a result calcium efflux is reduced, because when the sodium concentration is more, along with sodium, calcium ions are also there. So calcium will not move out and calcium concentration increases, because at other side we have other variety of pump. You can see here. This is sodium potassium pump, 3 sodium out, 2 potassium in. So sodium concentration decreases here. Okay, when sodium concentration decreases, what happens? There is sodium calcium counter transport, means sodium enters and calcium moves out. So when this is inhibited, this is also inhibited. This is secondary active transport. So primary is inhibited here, this is also inhibited. So calcium will not go out. Calcium remains inside, and that results in increase in the contractility of cardiac muscle. Another variety of pump, that is calcium pump. It causes transport of calcium ions from inside to outside, and it helps to maintain low concentration of calcium inside the cell. So it is commonly present in the sarcoplasmic reticulum. That is, we have discussed smooth endoplasmic reticulum of skeletal muscle. So what does it do? After muscle contraction is over in the skeletal muscle, this calcium ions are going back into the sarcoplasmic reticulum where it is stored. Okay, so here the concentration of calcium inside the cell is reduced, and it is also there in the cell membrane and some other cell organelle membrane. Then hydrogen pump, H plus pump. that is also present in many cell organelles, that is also present in H plus pump, that is also present in many cell organelles and it causes transport of H plus ion against the gradient. Then H plus K plus pump, that is present in the parietal cells of the gastric gland. In the stomach, H plus ions are secreted by this pump, okay? H plus K plus pump, and in kidney tubule also, kidney and stomach and kidney tubule. This is all about primary active transport. Now, next is secondary active transport. What is the secondary active? Active transport means against the gradient, two things are there. Against the gradient and energy is needed. But the thing here is against gradient, energy required. But what is the important difference here from primary active transport? Primary active transport energy comes directly from ATP. Here it will not come directly from the ATP. It is this energy has been stored in the form of concentration gradient somewhere else, and that is being used. For example, suppose here is the sodium potassium pump, 3 sodium out, 2 potassium in. Okay? So because of that, sodium concentration decreases and that causes transport of the other substances here. This is secondary active transport. This is again of two types, co-transport and counter-transport. Co-transport means both the substances are transported in same direction, and counter-transport they are transported in opposite direction. Now, for co-transport, here again also transport of substances in the same direction. Here also carrier protein is required, but the carrier protein here you can see, suppose this is a carrier protein. Sorry, I'll draw it like this. Suppose this is a carrier protein, but this carrier protein has two binding sites. One for, suppose if we are talking about co-transport, so there is sodium and glucose. One for sodium, other for glucose. When both of them are binding, then only there is transport. This is again active transport energy is required. So which are the examples of co-transport? Sodium, glucose, co-transport, sodium amino acid co-transport, sodium chloride co-transport, sodium iodide co-transport. So these are all the examples. This is counter-transport. Here the substances are transported in opposite direction, means one substance enters, other substance exits. And examples are sodium, potassium counter-transport. You can see here one positive, other positive. Here you can see one positive, one negative. So sodium potassium, then sodium hydrogen. This is very important. Sodium potassium counter transport is 1:1. If we are talking about the pump, that is 3:2. 3 sodium, 2 potassium. So this is different. This is pump is equal to primary. If you want to remember P for P, whenever you have the word pump, then it means primary active transport. And if the word is counter or co-transport, that is secondary active transport. So sodium potassium counter transport, sodium hydrogen counter transport, sodium calcium 1 positive counter transport, calcium potassium counter transport, calcium magnesium counter transport. Then here it is written calcium magnesium, calcium potassium. Then one important chloride shift, chloride bicarbonate counter transport. So these are all the examples of counter transport. So here we have completed simple diffusion or passive transport and active transport. In next session we will discuss about special categories of transport.
If you wish to get all topic notes, my book Physiology Notes is available on Flipkart as well as Amazon, and link for both that is given in the description box. And to get the soft copy, my application Vesalius that is available on Play Store, you can download it and get the subscription. Link for that is also given in the description box. Thank you so much.